Welcome to my course on Microbial Biotechnology. We are at Module 2 where we are discussing about structure and life cycle of representative groups of virus, prokaryotic and eukaryotic microorganisms. In the last lecture, we discussed in detail about the ultrastructure of bacteria and how the endospore formation takes place. In this lecture, number 4, we will be discussing about the diversity and structure of a few selected eukaryotic microorganisms. These are the contents of this lecture. We start with an introduction about the diversity of eukaryotic microorganisms. Then we will study about the ultrastructure of eukaryotic cells in the way we study about the ultrastructure of bacteria. Then we will discuss about algae, what are the characteristics, morphology, cell wall, pigments, then reproduction in algae, and then we move on to discuss about fungi, structure, morphology, then mold classification, and also finally about the protozoans, just dealing with the general characteristics of protozoa. So, eukaryotic microorganisms are a rich and diverse group within the domain eukarya, characterized by a true nucleus and membrane bound organelles that enable complex cellular processes. We have discussed about nucleoid, which is a false nucleus in bacteria, and we discussed about various organelles in the bacteria. So, in these eukaryotic microorganisms, we will be also discussing about them, which are different from the bacterial organelles. These eukaryotic microorganisms inhabit various environments and play important roles in nutrient cycling, primary production, and also in symbiotic relationships. Additionally, some of them are pathogenic, which causes disease in plants, animals, including humans. So, here we can see various types of eukaryotic microorganisms. For example, in A we can see the freshwater algae, Spirogyra and Zygnema. Then in B we can see Euglena, and then we can see the image of Saccharomyces cerevisiae in C, and we can see the image of the clonal sporangia of Rhizopus in the figure D. So, eukaryotic microorganisms are quite diverse, as we have already mentioned. In this figure, you can see a amoeba with pseudopodia, and let us begin with one of those types of eukaryotic microorganisms. For example, protozoa. These are single celled organisms which are either free living or parasitic. They are classified by their locomotion methods, that is amoeboid, for example, amoeba, flagellated trypanosoma, then ciliated paramecium, and sporozoum, which are the examples of plasmodium. Another type of eukaryotic microorganisms are the algae, and here you can see the chlorella under 1300x magnification. These you can see some green coloration here, and these are because algae are photosynthetic eukaryotic microorganisms ranging from single celled forms to multicellular ones. For example, the seaweeds, they are essential primary producers in aquatic ecosystems and significantly contribute to global oxygen production. So, apart from the trees, the algae are very important in production of oxygen, which helps in the survival of animals like us. Another example is that of fungi. In this picture, you can see Penicillium spinosum. Fungi are basically a diverse group of eukaryotic microorganisms, not one or two type, and it includes yeasts and molds. They play crucial roles as decomposers, breaking down organic matter, and can form symbiotic relationship with plants and algae. For example, mycorrhiza form symbiotic relations with plants, and lichens form symbiotic relationship with algae. Then there are other interesting types like the microscopic animals. You can see here Notalka, which is a rotifier. These eukaryotic microorganisms are classified as microscopic animals like nematodes, rotifiers, and they are small multicellular organisms found in various environments. Now, let us look into the ultrastructure of an eukaryotic cell. So, this is the structure of a typical eukaryotic cell with so many different well defined organelles like the mitochondrial, which is the powerhouse. Then this is the cytoplasm, which is basically the fluidic material, which includes all the different organelles as well as soluble proteins and other biochemical molecules. Then we have structures like lysosomes and a well defined nucleus, and then we have the plasma membrane and the ribosome, which is the protein translation machinery. We will discuss about these in detail in the next few slides. Let us now discuss about the various components of an eukaryotic cell. Let us start with the eukaryotic cell wall. Eukaryotic cell walls are rigid structures that surround the cell membranes of certain organisms. They differ from prokaryotic cell walls, which are primarily composed of peptidoglycan. The composition of eukaryotic cell walls varies widely among different groups. For example, fungi, their cell walls are primarily made of chitin along with polysaccharides and glycoproteins. Chitin provides rigidity and protection, through the exact composition can vary among different species. Algae, here the composition of the cell wall is again highly diverse. For example, diatoms have silica based walls. Red algae rhodophyta contain a mix of cellulose and sulfated polysaccharides. Brown algae phaeophyta possess alginates and other polysaccharides. Protozoa, some protozoa like amoebas have cell walls made of proteinaceous materials, while others such as paramecium lack rigid cell walls altogether. Major organelles, the nucleus. The nucleus contains the cell's genetic material and is key to DNA replication, transcription and regulating gene expression. The Golgi apparatus consists of flattened membrane bound sacs called cisternae and play a central role in intracellular transport and the post translational modification of proteins and lipids. So, here you can see a Golgi apparatus and the nucleus of a eukaryotic cell. The endoplasmic reticulum is an extensive membranous network connected to the nuclear membrane and closely associated with the Golgi apparatus. So, here you can see the nuclear membrane on the one side, and you can see the connection with the Golgi apparatus on the other side. Now, there are two types. One is the rough endoplasmic reticulum. These contains ribosomes on its cytosolic surface and is involved in protein synthesis, and there is the smooth endoplasmic reticulum. It functions in lipid metabolism, carbohydrate metabolism, drug detoxification and calcium ion storage. The next important organelle is the mitochondria. These are essential organelles in eukaryotic cells, known as the powerhouses of the cell for generating ATP through cellular respiration. They have an outer and inner membrane and contains their own small circular DNA. So, you can see the mitochondrial DNA over here, and then you can see the inner membrane, and then you can see the outer membrane and the intermembrane space between these two membranes. Another important structure is the chloroplast, not available in all kind of eukaryotic microorganisms. These chloroplasts are specialized organelles in plants, as we know, and also available in algal cells and are responsible for photosynthesis. They contain the green pigment chlorophyll, which captures sunlight for this process. Chloroplasts have a double membrane structure and possesses their own circular DNA, as in the case of mitochondria. So, you can see here certain structures called thylakoid, and there is a chloroplast envelope. Again, it has an outer membrane, a inner membrane, and a intermembrane space, and then you have these nucleoid, which are the DNA rings, and then we have ribosomes over here, and also it has the starch granules, which are basically produced as a result of the metabolic processes inside. One more important organelle is the lysosome. These are specialized single membrane organelles filled with digestive enzymes called hydrolysis, which break down and recycle cellular waste, including damaged organelles and foreign particles, and help maintain cellular homeostasis by preventing the accumulation of harmful substances. Centrosomes are small non-membrane bound organelles near the cell nucleus in animal cells. They serve as the main microtubule organizing centers, or MTOCs, and play critical role in cell division and organization. And then you can see here the centriole, the mother centriole, and these microtubules, and then here are the daughter centrioles. Other important organelles are the microbodies. These are single membrane organelles that contains enzymes for various metabolic pathways. The peroxisomes involved in fatty acid oxidation and detoxification of harmful substances like hydrogen peroxide. Then the glyoxysomes, which contain enzymes for fatty acid oxidation and sugar synthesis via gluconeogenesis. Then there are glycosomes, which house or contain glycolytic enzymes, and there are the hydrogenosomes, which contain enzymes for producing molecular hydrogen and ATP in anaerobic conditions. One of the important component of an eukaryotic cell or eukaryotic microbial cell is the cytoskeleton. This is a dynamic network of protein filaments, and it consists of three main types: the microfilaments, microtubules, and intermediate filaments. The smallest and most flexible microfilament are made up of actin proteins. These are involved in cell movement, division, and the maintenance of cell shape. The microtubules are larger and more rigid structures made of tubulin proteins and are crucial for intracellular transport, cell division, and also in the maintaining the shape of the cells. The intermediate filaments are intermediate in size, and these are composed of various proteins providing mechanical strength and structural integrity to cells and tissues. So, here you can see how an actin filament and a microtubule and an intermediate filament is helping in maintaining the structure of the cell overall. Like prokaryotic bacteria, eukaryotic microorganisms also may have flagella. These eukaryotic flagella are long whip-like organelles critical for movement, locomotion, cell signaling, and fluid transport. Their core structure consists of microtubules in a 9+2 arrangement with dynein motor proteins on the outer doublets, generating ATP dependent movement. Flagella originate from a basal body derived from the centriole, which anchors the flagellum to the cell's cytoskeleton. So, here is the basal body, as we can see, and then these are the cross section where you can see the plasma membrane and you can see the dyneins as we have mentioned in this lecture. Another important component is the cilia. These are short hair-like structure on the surface of many eukaryotic cells, similar to flagella but shorter and more numerous. Cilia have a core of microtubules, again arranged in a 9+2 pattern with dynein motor proteins for ATP dependent movement. They originate from a basal body derived from the centriole, anchoring them to the cell's cytoskeleton. Cilia play key roles in motility, sensory perception and fluid movement in eukaryotic organisms like protists, algae, fungi and also other eukaryotic microbial animals. Let us now discuss about the algae. In this picture you can see the image of a marine algae. The long hairy strands of that means rope are about 1 meter high and the bottom is covered with soft planked weed. Overall, algae are photosynthetic organisms with chlorophyll except colorless types like Prototheca. So, here you can see some of the freshwater algae diversity in a pond in Moscow recorded by Alexander Klepnev. Algae are polyphyletic, originating from different evolutionary lineages ranging from unicellular microalgae, example chlorella, Prototheca, diatoms, to large multicellular forms like giant kelp. Algal plastids responsible for photosynthesis originated from cyanobacteria through various endosymbiotic events. Green algae have primary chloroplast from cyanobacteria, while diatoms and brown algae have secondary chloroplast from red algae. Unlike terrestrial plants, algae are characterized by lack of specialized cells and tissues such as stomata, xylem and phloem, as well as structure like leaves, roots and rhizoids. Most algae are phototrophic, using photosynthesis, while some are mixotrophic, obtaining energy from both photosynthesis and organic matter. Certain unicellular algae like green algae, euglenids and dinoflagellates are heterotrophic and depend on external energy sources. Although called blue-green algae, cyanobacteria are excluded from algae as they are prokaryotes. Algal morphology, let us look into unicellular algae. Unicellular or acellular algae are single-celled organisms which may be either motile or non-motile and be classified into 3 forms: rhizopodial forms lacking a rigid cell wall. They have flexible shapes and move using pseudopodia. Some examples are Chrysamoeba, then Rhizochloris, Dinophyceae. The flagellated forms are the motile algae with flagella, example Chlamydomonas. Then there are coccoid forms, these are non-motile with a rigid cell wall and absence of flagella except in reproductive stages, example Plochloron and Ephanocapsa. So, here in this picture we can see various morphologies of unicellular algae. Here we can see Chrysamoeba, and then in B we can see Chlamydomonas, and then in C we can see Clostridium, and then in D we can see the chlorella. An algal colony is a group of similar cells held together by a mucilaginous envelope. So, some of the algae are unicellular as we have discussed, and they may exist independently, but in a algal colony they all exist together, which are held together by a mucilaginous envelope. One important concept here is that of conibium. A fixed number of cells arranged in a specific pattern is called a conibium, and these remain constant during growth, for example, Volvox and hydrodiction. Then the next is the palmelloid. The non-motile cells are embedded in a gelatinous matrix with a variable number, shape and size, each functioning independently, for example, palmella and gliochloris. So, here we have these Volvox aureus in figure A, then we have Phaeocystis antarctica in figure B, and then in figure C we have Hydrodiction reticulatum, and then in D we have palmella. So, these are different kinds of morphology of the algal colonies. In addition to these, we may have tetrasporial. Here these features pseudocilia made of mucilage. So, next comes the dendroid. These are cells connected in a branching tree like structure via the mucilage, and then we have the rhizopodial morphology, where the cells are united by the rhizopodia. So, in A we can see Palmodiction viridi, which is a tetrasporial morphology. Then we see in B the palmella, and then in C we see the dinobryon divergence, and in D we see Chysidiastrum catenatum, which is basically rhizopodial. Let us now study about another morphological form of algae, the filamentous algae. These filaments form through transverse vegetative divisions linking cells via the middle lamella. These can be unbranched or branched. Branching can be again true or false. False branching is common in cyanobacteria, while true branching occurs in various types of algae. This branching can be simple, which is an upright branched thallus attached by a disc from the basal cell, as in the case of Cladophora, you can see in figure A, or it can be heterotrichous, which is basically thallus with prostate base and an erect branch system, example Stegioclonium in the case of figure number B, or it can be parenchymatous, where cells of primary filament divide in all directions, losing the filamentous structure, as can be seen in figure C, and some of the examples of parenchymatous forms are the Porphyra, Alva, Enteromorpha. Algal cells exhibit a wide diversity in composition and structure, and in a very simple term you can see that it has a primary cell wall, then a middle lamella, and here on the other side you can see the plasma membrane and various components over here like pectin, cellulose microfibril, and as well as hemicellulose, and here we can see the presence of certain soluble proteins. Cellulose is the main structural component in most algae. Other polysaccharides like mannans in green algae and red algae like Porphyra and xylans also contribute to cell wall formation. Alginic acid and alginate salts are found in brown algae, silicon in diatom cells, and calcium carbonate in some tropical marine algae. Algae also has certain pigments, and as we know, the main pigment which is visible to us is the green pigment or the photosynthetic pigment, chlorophyll. These allow them to capture light energy for photosynthesis. The types and abundance of these pigments differ among algal groups, enabling adaptations to various light conditions and ecological niches. Specific pigments give algae distinct colors ranging from green to red, brown or blue green, depending on the composition and concentration. In general, algae contains several types of photosynthetic pigments. For example, it may have chlorophylls which are of 5 types A, B, C, D and E, or carotenes again of 5 types function as accessory pigments, including alpha, beta, gamma and so on, and also flavicin. Then we have xanthophylls. There are approximately around 20 different xanthophylls identified, and these are also characteristics of various algal groups and many times help in their identification. Then we have phycobilins, around 6 types, and these are also again specific to some algae. Let us now discuss about the reproductive systems or reproduction in process in algae. So, basically algae, one of the important reproduction method is through vegetative means. This involves a specialized part of the thallus which detach to form a genetically identical offspring without variation. So, this is true to type. It can occur through several mechanisms, for example, fragmentation. Filaments break into pieces, is capable of growing into a new thallus, as in the case of Eukaryotic Spirogyra. And then another method is by fission. A single cell divides through mitosis, forming 2 daughter cells common in desmids, diatoms and unicellular algae. Then we have adventitious branching. Here new branches, example Protonema in Chara, grow into new thalli when these are detached. Then another method is the tubers, which are spherical structures on Cara's nodes or rhizoids that develop into new thalli when detached. In some cases, algae also undergo budding, as in the case of Protosiphon, and this reproduce by forming new individuals by a process which we call as budding. Another method of reproduction is asexual reproduction. This asexual reproduction in algae occurs through spores formed within vegetative cells or specialized structures called sporangia. These single cell spores can develop into new thalli. The type of spores include hypnospores. These are thick-walled, non-flagellated spores produced under favorable conditions by some green algae like Chlamydomonas and Protosiphon. Then we have zoospores, as the name suggests. These are flagellated asexual spores formed in zoosporangia or directly from vegetative cells, example again in Chlamydomonas, Eukaryotic. And then we have aplanospores. These are non-flagellated, thin-walled spores formed when flagella formation fails under unfavorable conditions. Then we have tetraspores. These are non-motile spores formed in tetrads within tetrasporangia, example in the case of Rhodophyceae and Phaeophyceae. Algae also undergo sexual reproduction, which involves the fusion of different gametes, exhibiting a diverse range of characteristics and reproductive methods. Gametes can originate through mitotic divisions from any vegetative cell of the thallus, which acts as a gametangium, and specialized gametangia may form. When haploid gametes fuse, they create diploid zygotes, leading to the development of a new thallus. Sexual reproduction in algae can take several forms based on the morphological and physiological characteristics of the gametes. For example, it may be isogamy, anisogamy, oogamy, hologamy and autogamy. Isogamy is the fusion of morphologically similar but physiologically distinct gametes, positive and negative, example in the case of Chlamydomonas, Eukaryotic, Zygnema, Spirogyra. Anisogamy is the fusion of morphologically and physiologically different male microgametes and female macrogametes, example in the case of Chlamydomonas brownie. Then oogamy is the fusion of large female gametes with smaller male gametes, example Oedogonium, Vaucheria, Cara and Laminaria. Then we have hologamy, which is the fusion between opposite strain thalli where the entire thallus act as a gamete, example seen in Chlamydomonas. Autogamy is the fusion of two gametes of opposite strains from the same cell with no genetic recombination as in the case of diatoms. Now, let us discuss about algae which forms symbiotic relationships. Lichens are stable vegetative bodies formed by the association of a fungus and a photosynthetic symbiont, typically a species of green algae called phycobiont. So, in this file figure, we can see the lichen Cladonia, Portentosa and Hypogymnia physodason, a dead branch of a plum tree. In some coral forming invertebrates, dinoflagellates act as endosymbionts as using incident light and the host's carbon dioxide to produce sugars and oxygen. The loss of these dinoflagellates from the host leads to coral bleaching and reef deterioration. So, they are of huge ecological importance. So, here you can see the picture of coralline algae on undersides of an coral. Let us now discuss about the fungi. So, fungi are a diverse group of eukaryotic organisms including yeasts, molds and mushrooms, and they belong to different kingdoms separate from Plantae, Animalia, Protozoa and Chromista. As heterotrophs, fungi absorb nutrients by secreting digestive enzymes and cannot perform photosynthesis. They play a vital role as decomposer, aiding nutrients cycling in ecosystems. Fungi also form symbiotic relationship with plants like mycorrhiza, animals or other fungi and can act as parasites in various environments. So, in this panel we can see in figure A the Candida albicans here, and then in B we can see the Penicillium species, and in C we can see Asci or ascospores of Morcella elata, and in D we can see Tarantula which is infected by endopathogenic fungi, Ophiocordyceps, and this is actually also economically very important. And then in E we can see the microscopic image of Aspergillus niger. So, these are the diverse types of fungi in brief, but there are many more which cannot be shown in one single slide. The structure of the fungal cell wall vary in composition and structure across species, affecting their elasticity, permeability and pathogenicity. Typically, they have a layered organization, an inner layer of chitin and beta glucans. These provide shape and strength, and an outer layer of monones and glycoproteins which influence properties like hydrophobicity, adhesiveness and immune response. And there are also certain associated proteins in this cell wall. The composition of the fungal cell wall, it has several key components that vary across species. We already discussed about chitin. It is a polymer of an acetyl glucosamine which provides structural rigidity. Then we have the beta glucans. These are glucose polymers with beta 1,3 glucans forming the backbone and beta 1,6 side chains. Then we have the monones. These are polysaccharides of mannose units forming part of the outer layer, often associated with proteins, and associated proteins which include enzymes, adhesins and glycoproteins contributing to various cell wall functions. Let us now discuss about yeast, which are eukaryotic single celled microscopic fungi. So, in this picture you can see Saccharomyces cerevisiae in figure A. Then you can see Candida albicans which is grown on suberwood agar. Then in C we can see the baker's yeast, and in D we can see yeast and bacteria from symbiotic culture of bacteria in yeast or SCOBY. There are over 1500 yeast species identified, and this represent only about 1% of all known fungi. Though unicellular, some yeast evolved from multicellular ancestors and can form pseudohyphae. Yeast sizes typically range from 3 to 4 micrometer in diameter. While yeast is often associated with Saccharomyces cerevisiae, they are phylogenetically diverse and belong to two distinct phyla, Ascomycota and Basidiomycota. In most yeast, the common morphotype involves one nucleus per cell. However, Asbia gossypi displays a syncytium where multiple nuclei share a single cytoplasm, each following its own replication cycle. In Toxoplasma gondii, endobudding occurs where daughter cells form inside the mother cell after nuclear division, leading to distinct plasma membranes for each nucleus and is linked to spore formation. Moles. These include a wide range of fungal species with diverse strategies such as saprotrophs, mesophiles, psychrophiles and thermophiles. Some moles can pose opportunistic health risk to humans. Moles, like other fungi, are heterotrophic and do not perform photosynthesis. They secrete hydrolytic enzymes primarily from hyphal tips to break down complex biopolymers like starch, cellulose, lignin into simpler compounds for absorption, playing a key role in organic matter decomposition and nutrient recycling in ecosystems. In this picture, we can see a fruit mole growing on strawberry, and then in B we can see fruit mole stained with methylene blue under 2000x magnification. The morphology of moles. Moles are characterized by filamentous structures called hyphae, which grows into a network known as mycelium, with each hypha about 5 to 10 micrometer wide. In some fungi, hyphae are divided by septa with central pores allowing cytoplasmic flow. Fungi exhibit three hyphal types: sunocytic, these are non-septated, then septate with uninucleated cells and septate with multinucleated cells. Asexual spores form at the hyphal ends, giving moles their characteristic dusty appearance. So, these are the three type of hyphae, which is sunocytic, and then septate, and then septate with multinucleated cells. So, here you can see several nucleus in one cell, and here there is no any septa. How moles are classified? Moles are classified as microbes, but do not belong to a specific taxonomic group, primarily found in the divisions Zygomycota and Ascomycota. Many moles were formerly classified as Deuteromycota. Zygomycota or conjugated fungi feature non-segmented hyphae with multiple nuclei, which fuse during sexual reproduction to form zygospores. In contrast, Ascomycota have septate hyphae with a single nucleus per cell and produce spores in sac-like structures called asci during sexual reproduction. Zygomycota. Zygomycota is a classification within the kingdom of fungi, which encompasses over 1060 species from the groups Mucoromycota and Zoophagomycota. This group is characterized by the formation of zygosporangia during sexual reproduction, where resistant spores are produced. A unique feature of Zygomycota is the presence of chitosan in the cell walls instead of the more common chitin. Many Zygomycota are utilized in various industries to produce compounds such as biotin, lipases, arachidonic acid, linoleic acid and carotenes, and are therefore very important from the point of view of microbial biotechnology. In figure A here we can see rhizopus with an immature zygosporangium, and then in figure B we can see sporangia of a mucorales fungi growing on a pitch. The morphology of zygomycetes. These are typical mole structures along with specific features, and you can see this typical structure of the Zygomycota, where you have this sporangium, then columella, and then the rhizoid, stolon, and sporangiophore. The rhizoids are root-like structures that help the thallus adhere to the substrate and increase nutrient absorption. Sporangia are fruiting bodies that contain spores of various shapes and sizes. Sporangiophore is a specialized structure that bears the sporangium. Columella, the dome-shaped tip of a sporangiophore that causes the sporangium to rupture and release spores under suitable conditions. Then we have the stolon, which is horizontal hyphae that grows along the substrate. Let us look into the reproduction in zygomycetes, the asexual reproduction. Zygomycetes produce two types of asexual spores, the mitospores and chlamydospores. Mitospores, also known as sporangiospores, are produced asexually in specialized structures called mitosporangia. These are carried by hyphae known as mitosporangiophores. Sporangia exhibit negative gravitropism and positive phototropism to facilitate spore dispersion. Mitospores have walls containing sporopollenin, making them highly resistant to degradation. Chlamydospores are spores primarily aiding in mycelium persistence and are released when mycelia degrade. They have thick pigmented cell walls and lack dispersal mechanisms. Let us now discuss about Ascomycota. Ascomycota or sac fungi is the largest phylum of fungi comprising over 64,000 species. The defining feature of this group is the ascus, a microscopic sexual structure that produces non-motile spores called ascospores. Family examples include morels, truffles, and yeast used in baking and brewing. Ascomycetes are crucial for humans, providing antibiotics, aiding in fermentation of bread and beverages, and serving as model organism in research, for example, Neurospora crassa and various yeasts. In this figure, we can see some of the species of mold which belong to Ascomycota. The morphology of Ascomycetes. So, here we can see the presence of hyphae, which is again running parallel to the substrate. Then we have a food cell from which the stalk emerges, and then we have a vesicle, and then we have the chains of conidia. So, together this entire structure forms the conidiophore. Ascomycetes share typical mold structures, but they also feature conidiophores, which are specialized hyphae that produce asexual spores called conidia. Common features of conidiophores include, as already discussed in this figure, stalk, which is a cylindrical or elongated stalk, then vesicle, a swollen or flask-shaped structure at the apex of the stalk where conidia form, then conidiogenous cells. These are cells on the vesicle that generate conidia, and then finally conidia, which are asexual spores produced by mitotic divisions in their conidiogenous cells. Asexual reproduction is the primary method of propagation in Ascomycota, primarily through the production of vegetative spores called conidia. Conidia are single nucleus spores resulting from mitotic divisions, making them genetically identical to the mycelium from which they originate. So, they are also true to type. Depending on the species, conidia can disperse via wind, water, or animals. Various types of conidia can be distinguished by color, shape, and release methods, serving as taxonomic characteristics for classifying Ascomycota. Let us now discuss about protozoans in brief. Protozoa is a subkingdom of Protista with over 50,000 described species. They are diverse single-celled eukaryotes found in nearly every habitat, including freshwater, marine, and soil environments. These organisms exhibit various forms of locomotion, such as flagella, cilia, or pseudopodia. Most humans harbor protozoa which can be commensal or pathogenic. While some protozoa coexist harmoniously with their host without causing harm, Others can lead to diseases ranging from mild infections to life-threatening conditions. This dual nature underscores their ecological significance as both crucial components of food webs and potential health threats in human and animal. In this panel, we can see various protozoans showcasing the structural diversity. You can see euglena in figure A, then we see trypanosoma in figure B, which is taken from the blood smear of patients with African trypanosomiasis, which is a disease. And we have mentioned that some of these causes disease in humans. Then you have the amoeba proteus with pseudopodia in figure number C, and Paramecium aurelia in figure number D. So, protozoans are quite diverse, as we have discussed and as we can see from this panel. So, let us discuss about some of the general characteristics of protozoa. Most protozoa are small, typically under 15 micrometer, and as unicellular eukaryotes, they have a membrane bound nucleus and organelles. The plasma membrane extends over locomotory structures like pseudopodia, cilia or flagella. Some protozoa have a pellicle that maintains shape while allowing flexibility. Their cytoplasm is often divided into ectoplasm, which is the outer layer, and endoplasm, inner layer, which contains the organelles. Certain protozoa also possess a cytosome or cell mount for ingesting particles. So, here in this figure we can see various protozoans, once again showcasing the various structural diversity as we have discussed in the earlier slide. So, with this we can see that protozoa is actually quite diverse and also very unique, particularly being a eukaryotic microorganism. Most protozoa, unlike plants, fungi and algae, lack a rigid cell wall. That is one of the unique features. In some, such as ciliates and euglenozoans, the outer membrane is reinforced by a flexible cytoskeletal structure called the pellicle, which aids in movement and maintains shape. Pellicles vary in composition. Ciliates and epicomplexa have alveoli supported pellicles, while euglenids have protein strips along the cell. Protists like euglenoids and Paramecium have pellicles in some cases. The pellicle may host epibiotic bacteria attached via fibria. So, with this we come to end of this lecture. So, we will be discussing about the various eukaryotic microorganism in the next lecture. Thank you.